Thank you very much, everyone. It's a real pleasure to be here at this meeting. That's always extremely motivational for me and my team in Edinburgh. And I'm delighted to give you a bit of an update on the work we've been doing in our lab. So I'm going to talk about IDH mutations, which, as you have heard of many talks already from this meeting, are one of the most common mutations in the intrahepatic form of cholangiocarcinoma, similar to FGFR mutations, seen in, depending which series you look at, between 10 and 15% of patients, and probably more if you combine IDH 1 and 2 together. This is probably the biggest published series of patients by the German group, 6,000 patients, and you can see that they co-occur with different mutations compared to the other targetable drivers. So particularly regulators of the epigenome, so less of the common drivers that we're used to, such as KRAS. So they commonly co-occur with ARID1A and BAP1, for example. So people have said to me, including at this meeting, why are we studying IDH? We know everything there is to know about IDH. We have an inhibitor. So hopefully I'll persuade you that we've only really scratched the surface in terms of what we know about these mutations and their mechanism. We, of course, have drugs in the clinic which are working very well, and you've heard them described very elegantly by John. And the classical description, so these are very special cancers, IDH mutated cancers. They're one of very few cancers ever in man to have described to produce something called an oncometabolite, so a product that occurs in cancer cells that is not found in healthy cells. And these mutations are relatively recently described, just in 2009, first in the brain in low-grade glioma. And they are thought to act in a special way where the IDH enzyme, which is expressed in all cells of the body and usually acts to catalyze a specific biochemical reaction as our cells metabolize. So they catalyze this reaction that some of the medics in the room may remember from their medical school biochemistry days, this enzymatic reaction from isocitrate to alpha-ketoglutarate. When the protein is mutated, this reaction cannot happen, and this new product called 2HG or D2HG is formed and accumulates in the cell. And the original description thought that this new product acts to competitively inhibit other enzymes imperative in the cell for things like DNA methylation. So methylation of DNA occurs to help us turn on and off genes within the cell, and it was thought that D2HG inhibited some of these enzymes and resulted in methylation of DNA. And what do we know already in cholangio specifically? Well, almost all of this work has been done by the amazing Bardisi lab in Boston in the States, and in particular these two papers where he's shown that IDH mutations have effects on cell identity and the blockage of differentiation of liver cells. So when he expresses IDH mutation in some of the albumin-expressing cells in the liver, they are blocked from becoming true differentiated hepatocytes and upregulate a lot of the progenitor genes in the liver. And in addition to that, he's shown very eloquently that this also has effects on the immune system. IDH mutations are thought to be what we call immune cold, so they don't have the cytotoxic T cells that are commonly found in other types of cholangiocarcinoma, and that probably this is done through repression of various genes, including interferon gamma receptor suppression. But there remain so many unanswered questions. We don't know why these cancers only arise in certain tissue types, so brain, the liver, and cartilage chondrosarcoma are the classic ones, as well as leukemia. And that tissue specificity and why those mutations happen in those tissue types is completely unknown. If we look at the data from cholangio, there's one of these mutations called R132C is very dominant in cholangiocarcinoma. We don't know why that is. And so there's lots, and the mechanism really still remains to be completely unpicked. So it's important that we have good preclinical models to answer all these mechanistic questions, and then we can start to address things like resistance to IDH inhibitors, occurrence of IDH2 mutations, and all the rest of it. So we set out to start answering these questions in the lab, first using cell lines and then new models in mice. So we used a technology many of you would have heard of, known as CRISPR-Cas9, to gene edit human cell lines taken from cholangiocarcinoma patients to introduce IDH mutations. So this is data looking at the RNA from these cells, and what we're looking for is a difference between the control cells, which are denoted here with the gray strips, and the mutated cells. So we have gene edited various of the mutations, IDH1 and 2, into the cells, and then looked at the whole RNA transcriptome. And you're basically looking for differences in the color signature of those cells. And so you can see just with the introduction of this mutation, which very often is just a single base pair change of DNA, that the cells get completely reprogrammed in terms of their RNA transcription. And when we look at actually what genes are differentially expressed in response to IDH mutation, we see very common signature across the different mutations, suggesting this may well be a common change, perhaps mediated through D2HG. And so for the first time, and this has never been done before by any lab, we've been able to compare the different isoforms and the different IDH mutations. So we're able to see which genes are common to IDH1 and 2, so actually very few genes, just 30 genes, and which are differentially expressed by the different mutations. And interestingly, R132C and IDH2 R140Q do tend to have specific transcriptional effects. And we were surprised in that we found quite a strong signature. I'm just going to go back a slide. So if we look at the right-hand side of the graphs, we see upregulation of a number of genes known as the HOXA genes. These are embryonically expressed genes. They're not classically involved in embryogenesis of the liver, but in all four mutations, we see upregulation of HOXA2, HOXA3, HOXA4, HOXA5, and also some of the intergenic long non-coding RNAs, one appropriately named HOTAIR M1. But so the whole HOXA cluster seems to be activated or indeed de-repressed, we don't know yet, by IDH mutation in this model. So we then looked at various pathways and how are these differences in transcription of genes actually affecting the phenotype and the functions of the cell. And actually the only mutation that came up positive on any kind of pathway analysis was R132C in the liver. So this is the one that we know is often mutated in intrahepatic cholangio. And it was really nice that we saw downregulation of interferon gamma, interferon alpha, and EMT. So that's validating all of Nabil's lovely work that he's published before. So it was nice to know that hopefully our model is working as expected. When we look at liver-specific genes and liver identity genes, we see upregulation in a lot of the progenitor genes or biliary genes in the liver, particularly those in the Notch pathway and some of the cytokeratins that are expressed on biliary and progenitor cells, as would be expected by the hypothesis as well. So we then looked at the metabolome. We just heard some metabolic data, but this is showing that, again, the mutations are clustering together compared to the CRISPR control lines. So these are metabolically being reprogrammed, these cells as well. And we showed using mass spectroscopy, again with collaboration with our colleagues in Japan and Kyoto, that they do produce the oncometabolite D2HG, which is produced at hugely high levels. And it's the R132C mutation, which seems to be the biggest D2HG producer. And that's also in line with the literature we know about in brain glioma. And we see quite interestingly differences between IDH1 and 2. So this is a very busy slide, but essentially what it's showing is all the metabolites that are either up-regulated or down-regulated in response to the mutation. And unlike many cancer cells where you see this thing called a Warburg effect, where the cancer cells become reliant on glycolysis in the cytoplasm, we actually see the opposite effect in IDH1. So we see a downregulation of lactate, and they're becoming less reliant on cytoplasmic glycolysis, and they're actually moving towards more of an oxidative phosphorylation type metabolism and reliance on things like fatty acids and amino acids in the cell. But we don't see that effect in IDH2. And that is likely due to the different location of these proteins within the cell. So IDH2 are mitochondrial enzymes, IDH1 are found in the cytoplasm. And so more work's needed, but we see this very different metabolic signature in response to IDH1 versus IDH2. So lastly, we went on to look at the methylation profile of the DNA in these cells, according to what they've seen in glioma. This is just a graph showing that we can that the mutations all cluster together in terms of the methylation profiles compared to other cell lines. But we see, again, a complete huge change in terms of the differentially methylated regions of the DNA in these mutations. And unlike what we were expecting, we thought we were going to see complete hypermethylation of the DNA. We see both hypermethylated regions, but also hypomethylated CPG islands. And again, the IDH1 mutations seem to be very similar. There's no significant difference between the DMRs in IDH1 and 2, but there's huge differences between IDH1 and 2 in terms of the methylation effects. And we then looked at where these islands, where the methylation is occurring. So if genes themselves are methylated, or if the promoters of those genes, which affects gene expression, are differentially methylated. And often promoters and enhancers of genes lie in what we call the CPG shores and CPG shelves. And we see, if you can see that these lines separate at the right end extreme of these graphs, showing that there's differences between the mutations in terms of where the DNA is being methylated. So this is all great in cell lines. We were surprised that we saw such a striking phenotype. And the main bulk of our work is trying to develop a really robust mouse model to study this in the in vivo context, in the natural microenvironment of the tumor. So we, in collaboration with Ian Tomlinson, geneticist at Oxford, we've been developing this, what we call a flex mouse, to express the mutation in mice. So we've designed this construct in which the IDH mutation is inserted into the genome in an antisense direction. So it's in the opposite direction that you would need for expression. When you give these mice a drug called tamoxifen, it activates these sets of DNA called LOXP sites, and it results in a 180-degree switch of the mutation, which means it's now in the correct orientation to be expressed. And you then get further activation of other LOXP sites, excision of all the other genetic machinery you need in the construct to make it work. And then finally, once the gene gets expressed, you ended up with a very clean system. And just like in human patients, these mutations are in the heterozygous form. So we've left the mice with one wild-type allele of IDH1 or 2 and one mutant allele. So we're as closely possible, modeling what we see in patients who carry IDH mutations. We then had to make the mice, so this construct is targeted into embryonic stem cells. The embryonic stem cells then get injected as blastocysts into mice, and we generate firstly chimeras of mice that are carrying the mutation, and then we generate a pure line. And the last couple of years of work have been characterizing these mice to ensure that this whole insert is has been put in the correct fashion with no mistakes and errors in the DNA, and that the mutation is being expressed. So this is just a PCR where we've amplified the DNA across the mutated exon, and you can see through this Sanger sequencing trace that the blue and the green peaks show the wild type allele, and these little red peaks here are showing that the mutation is present in the liver cells. And just to say also that this is a human codon, okay, so although we've knocked it into the mouse genome, it is the human IDH. If this is just looking at the same thing in a different way, where we've used something called a restriction digest to cut the DNA and to then sequence the cut area of the DNA that's expressing the mutation, and we're expecting the mutation to look like red, black, red on our Sanger trace, and so we're seeing the mutant very nicely in our mouse. So the next part of the work is all ongoing. We're now characterizing the mice. So we have two IDH1 mutations, two IDH2 mutations. It's really unknown in human disease which cells in the liver these mutations are arising in. The work that Nabil has done has used albumin-expressing cells in the liver, and so we're doing some work where we've put this mutation into both the cholangiocytes using a keratin 19 driver, so a classical driver of the biliary epithelium, but also in the hepatocytes, particularly the periportal hepatocytes that seem to proliferate and can differentiate during liver injury. So we've used this GLS2-CRE where we're expressing this in the hepatocytes around the portal tracts to see if these cells can de-differentiate and become biliary tumors. So we are using a fluorescent reporter known as a tomato or red fluorescent reporter to FACS sort these cells out of these mice, and to do all the analyses that we've been doing on the human cell lines. So we're characterizing them again using the transcriptome, the methylation arrays, and the metabolic mass spectroscopy assays. We've bred the mice to the other drivers that we see inherently in patients, so ARID1A, BAP1, and KRAS. And hopefully, the next time we meet, I'll be able to give you updates on this to see if some of the phenotypes we've seen in the cell lines, particularly the HOXA transcription, is this just a correlative effect that we see in response to D2HG and IDH mutation, or is this actually a biological mechanism that's producing this phenotype? We also want to see whether the metabolic reprogramming through what we call anaplerosis, which is like replenishment of the TCA cycle with its intermediates, whether we also see that in the mice. And what are the differences between the IDH1 and 2 mutant alleles, and can these be targeted for therapy? And with that, I'll stop and just to say a big thank you to everyone who's supported this work. Mouse work is not easy, and particular thank you to the AMMF for funding us at a real critical point in our journey, and a big thank you to Caitlin McCaffrey, my RA and lab manager in the lab, who I've just enjoyed working with so much. Thank you very much.